we were talking about the respiratory system. We looked about pulmonary ventilation. We talked about also external respiration, transport of gases, and then leading up to internal respiration. But really, before we can understand external or internal respiration, we need to more fully understand those pressure gradients that exist for each of the gases. And we didn't add any values yesterday, but we said, well, if we know where a gas needs to go, we probably know where it's in greatest concentration or pressure versus where it's in least pressure in the body. And so oxygen, we learned, is in greatest pressure where? It's greatest in the atmosphere, and the pressure gradient drops so that it's least where? In our tissues. And we said, why? Why is the pressure the least for oxygen in the tissues? If it's going in the tissues, why is it lower in the tissues? Why is oxygen content so low in our tissues? Why is oxygen content low in our tissues? Or not, we're saying, well, why is it present but in low concentrations in the tissue? So the pressure gradient does allow for its transport, and we're trying to understand better that pressure gradient. Why is the pressure higher in the atmosphere yet so low in the tissues? What is it about our body that makes oxygen content so low in the tissues to drive that pressure gradient and transport of the gas?
It's being used in the cell.
Very good. It's being used. Okay. As soon as it goes into the cell, we said it's used at the end of the electron transport chain and then converted to water. Okay. So as soon as it goes in, it does its work, and we need more to come in, pick up electrons. Okay. So it's so low in our tissues because, as correctly stated, it's being used, and that's why we want it. We're using it, so we better drop off more. So we need a pressure gradient. Does that make sense? Okay. So oxygen would always move down its pressure gradient to the area in which it's needed in our tissues, because that's where it's being utilized. Conversely, we know that carbon dioxide has the greatest pressure where? It's in the tissues. Why is the carbon dioxide pressure gradient greatest in the tissues? Why is the pressure greater in our tissues than, say, our alveoli or the atmosphere? Why is the pressure of carbon dioxide higher in our tissues than anywhere else? It's a byproduct, so we're actively making it. Okay. High carbon dioxide is good. That indicates cells are metabolizing. Okay. It's a byproduct. We want to make it, that's good, but we want to keep it in our tissues, right? So we've got to get it out. And so there's a gradient that's established, well, for the body to let it diffuse out. And so what we concluded with yesterday is that oxygen will diffuse down its pressure gradient where it's the greatest in our atmosphere and the least inside of your respiring tissues. We also learned the gradient is reversed for carbon dioxide. We can actually add values here, like you did from your handout number 6, which you submitted last week. We know the pressure within the alveoli of oxygen is about how many millimeters of mercury? In our alveoli, it's about 100 millimeters of mercury. So within your alveoli, the pressure of oxygen is about 100 millimeters of mercury. In our tissues, the pressure of oxygen is about 40. Very good. It's about 40 millimeters of mercury. So you have a gradient of how much? What's the difference? 60. 100. 60, 64, 104. Very close. Very good. So in other words, oxygen diffuses from the greatest pressure of about 100 millimeters of mercury in the alveoli down this pressure gradient into the blood. We'll talk about that in a minute. And eventually from the blood into our tissues, where the pressure is about 40 millimeters of mercury. You see how the oxygen moves from the greater to the lesser? What were the values for carbon dioxide that you found for handout 6? And actually I wrote on the board. Carbon dioxide pressure is how much in our tissues? How many millimeters of mercury? A little bit, 46, okay. In the tissues. And how much in our alveoli? About 40. So a gradient of only 6, much less than oxygen. But still a gradient. The gas would diffuse down its pressure gradient. Building on your discussion from yesterday, why do you think there's such a big difference in pressure gradients between oxygen and carbon dioxide? Why do you think it's 60 for oxygen, yet only 6 for carbon dioxide? What did we learn about those two gases yesterday? Which one is more soluble? Oxygen is less soluble. Okay, so carbon dioxide is more soluble. The one that's less soluble needs a bigger gradient to push it, right, from one spot to another. Very good. The next bit of text in your handout or your guided notes is actually going to just restate these values. Okay, looking at the pulmonary circuit. If we're looking at the pulmonary capillaries, where blood is entering in the lungs, our oxygen content is what? Is what? Oxygen, as the blood's going to the alveoli, oxygen content initially is low as it enters the alveoli, and our carbon dioxide is high. Okay? And so if we look at the partial pressure of oxygen as blood enters the pulmonary capillaries, the pressure in the blood is low at about 40 millimeters of mercury. Yet in the alveoli, we said that we had that 100, 104 millimeters of mercury in the alveoli. So we have a pressure gradient between the alveolus and the pulmonary capillary. Which direction would the oxygen flow between these two areas? From which region to which? From the alveolus into the capillaries. It's being pushed down its pressure gradient. We're going to add then, well, what happens to the oxygen? We know it probably goes and binds to what? Hemoglobin, right? Okay, most of it's probably going to go to hemoglobin. But once it goes in the blood, right, we've increased our blood content of oxygen. But we first see a pressure gradient between the alveolus and the capillaries to add oxygen to the pulmonary capillaries. All right, so we allow for the diffusion, right? We said greater, right, to lesser pressure. We also noted that gradient of about 60 to 64 millimeters of mercury is huge to allow for oxygen to load into the blood really rapidly. So remember, our blood is trying to get from point A to point B. So it's moving through our capillaries at a pretty steady rate. Therefore, we've got to keep the blood going, so we've got to push that oxygen into the capillaries with that huge gradient. So it allows for just blood flow to keep going. Does that make sense? It's not the blood's not like, hold on, right, wait for oxygen to hop on, right? So it's just the oxygen's being pushed into the pulmonary capillaries. At the same spot, at the same location, carbon dioxide's pressure gradient is reversed and much less when compared to oxygen. In the pulmonary capillaries, we learned the pressure of carbon dioxide is a little bit higher at about 46 millimeters, yet in the alveoli, about 40 millimeters of mercury. We can see the directionality of the gradient is reversed, which makes sense. We want to get carbon dioxide out of the capillaries into the alveolus. But we noted the gradient is less because we said carbon dioxide is much more soluble than oxygen in that liquid plasma, so it doesn't have to be pushed as hard. to move from one spot to another. And when we do that, we get the adequate ratio of oxygen in, carbon dioxide out, okay, with these difference in pressure gradients. Does that make some sense? A little bit? Any questions at this point? All right. Once oxygen's added to the blood, we'll add in just a minute, it'll be bound to hemoglobin or soluble in the plasma, and it's transported to the left side of the heart, then out to the systemic circuit, right, and then it goes to your systemic capillaries, okay, to serve your tissues, your brain, whatever, you name it. What do you think happens to the gradients? Are they still there? They're switched. So the oxygen moves from the blood into a tissue, and carbon dioxide moves from the tissue back into the blood. Okay, so they're just reversed. The values of factors are the same, still that about 60 millimeters of mercury, and for oxygen gradient, and about 6 millimeters of mercury for carbon dioxide. As long as these gradients are established, the gases will move where they need to go in the body. What questions do you have about this gradient, why it exists, how our body utilizes it, or why the values are different in different regions of our body? Well, what I'd like to address now, which we've kind of talked about, is, well, how do those gases actually get transported in the blood? We have an idea, but we'll just kind of formally address them. We know that oxygen is most preferentially bound to hemoglobin inside of our red blood cell, because we learned it's not very soluble in water, and we know things that aren't very hydrophilic need a carrier. Well, the carrier for oxygen is hemoglobin. It's not completely water repellent, if you will, so a small amount of it, of oxygen, can just go in the blood plasma, not bound to hemoglobin, and still be transported. So it doesn't have to always be bound to hemoglobin. If you remember, hemoglobin can carry up to 4 oxygen atoms, hopefully reversibly, meaning it binds and unbinds, right, lets it go. When hemoglobin is bound with oxygen, you can write HbO2, or you can call it oxyhemoglobin. That would be the proper term for that new molecule. If hemoglobin reversibly binds oxygen and releases it, hemoglobin becomes reduced by binding a hydrogen atom. If you remember, we said hemoglobin can bind gases and hydrogen atom, and so that's what it can do. We can see the reversible process. Reduce hemoglobin in the presence of oxygen, can bind it, and releasing hydrogen atom into the solution. But it's very much reversible, so it's just kind of like a shuttle or a taxi, if you will, for oxygen. If we look at how hemoglobin works, just because it can carry 4 oxygen atoms, is it guaranteed that it always does? No, we can already see here it can carry hydrogen ion, it can carry carbon dioxide, it can carry carbon monoxide, whatever. If a particular hemoglobin molecule is carrying strictly oxygen and all 4 binding sites are bound with oxygen, that particular hemoglobin molecule is considered to be saturated. Okay? If hemoglobin is saturated with oxygen, all 4 binding sites are fully bound with oxygen. So it can be saturated or not, okay, or partially saturated. It's either fully saturated with 4 oxygen atoms or partially saturated with 3 or less oxygen atoms in a particular hemoglobin molecule itself. Okay. We're aware that hemoglobin in our red blood cells is there to bind and unbind oxygen and transport it. And I think we had a question earlier in the semester, I think it was in the first unit. I faintly remember it. Someone said, well, how does oxygen know to go to the tissue? Or maybe it was a a discussion about the steroid hormones, I think is where it actually came. How does the steroid hormone know to let go of its carrier? It doesn't inherently know, but as our blood goes through our tissues, local conditions change. And then what changes? Conditions, factors change, and then what changes? We could say the what changes. The function of something changes, right? And so as blood goes to our tissues, obviously our carbon dioxide content is going to change, oxygen content is going to change, temperature is slightly different, pH is different. And there's other factors here as well, but these are the big ones that kind of resonate with which we've covered this semester. So if you think about blood going to a capillary, say in your skeletal muscle after you're exercising, you're in the gym, right? You're running. What's happening to the tissues in your leg? To the skeletal muscles. What are they doing? They're taking in oxygen. They need oxygen. Okay, what else? So the oxygen content in that cell is beginning to go. Well, first it decreases, right? It's using all that oxygen. What else is happening? Very good. So oxygen's going down. Carbon dioxide production should go up, becoming more metabolically active. What else is going on? Skeletal muscle, we said, bulk of its energy is transformed to. There is some lactic acid maybe being accumulated, but the bulk of our energy transfer is in the form of heat. So temperature begins to rise, increase. What do you think about pH? I'm becoming active. It's declining, okay? Especially if you're making a lactic acid. But if you remember, carbon dioxide accumulates, carries out a reaction, and we produce hydrogen ion. So all of those factors in metabolically active tissue actually change the environment so the hemoglobin unbinds oxygen, which is good because when oxygen is dropped off, the tissue can continue to metabolize and do its work. And so let's look at this. If I want to release oxygen, my pressure for oxygen must probably be a little bit. If I want to drop it off to an area that area should have the pressure of oxygen should be actually be low because it's it needs it, right? So low partial pressure of oxygen. What about partial pressure of carbon dioxide? Beginning to what? Which way? Increase. Okay. So high carbon dioxide, low oxygen favors hemoglobin to let go. If these things are going on, our temperature is probably up. And our pH is probably becoming, we said, a little bit acidic. It's decreasing and becoming a little bit acidic. So in other words, a metabolically active tissue who's naturally going to have lower oxygen, higher carbon dioxide, higher temperature, slightly lower pH, actually favor hemoglobin to release what? What are we releasing? Oxygen. Very good. And that's summarized here. We said as cells become metabolically active, carbon dioxide is released, causing that increased partial pressure of carbon dioxide. I have here an increase in hydrogen ion content, which we know brings your pH which way? Second point. Your hydrogen ion content goes up, pH goes down and becomes acidic, which causes hemoglobin to let go, if you will, of the oxygen. Heat also promotes the release of oxygen from hemoglobin. These collectively, and other factors too, this isn't all inclusive, make sure that oxygen is unloaded to those tissues that need it. There's no need to drop off oxygen to a tissue that doesn't need it more than a tissue that does need it. So it's just kind of interesting how an active tissue can ensure its delivery of oxygen, right, to that particular tissue. So metabolically active tissue, the factors change to allow for oxygen to unload to that tissue. What are the two ways which oxygen can be carried in our body? bound to hemoglobin or just dissolved in the plasma as molecular oxygen. What about carbon dioxide? How can we transport carbon dioxide? Dissolved as carbon dioxide. It can also go into hemoglobin. Okay, so it can be dissolved, it can bind to hemoglobin. And what else did you learn from lab about carbon dioxide? High proportion of carbon dioxide is not bound to hemoglobin nor dissolved. The bulk of it is converted to bicarbonate ion. Okay? And that's what we're going to summarize and review here. We said a small percent, like maybe 10% of your carbon dioxide, can just be transported in the blood plasma as molecular carbon dioxide. We also know that about a quarter of your hemoglobin can actually restate that, about a quarter of your carbon dioxide can be carried in the red blood cell, bound to hemoglobin. In that case, you call it carbaminohemoglobin. The proper name for that. But we also know that the bulk of the carbon dioxide undergoes a transformation to form bicarbonate ion. And maybe this is a good point to put maybe a subset 3A, 3B to reiterate the locations in which this could happen. What were the locations where this could happen? From pre-lab that you guys did last week. It said bicarbonate ion can be formed in one of two locations, either one of two locations. They're actually on the board here. Where are we? Where in the body are we? In the blood. Okay, so where are the two locations you could be in the blood? Either in the plasma or in a red blood cell, right? Okay. So carbon dioxide can undergo this transformation in either of those locations. We said in the plasma it occurs how? In the plasma, if bicarbonate ion is formed, it occurs in what fashion? What term did we use? What was the difference in the location? Plasma versus red blood cell. Worksheet 11. Probably that's not where a high proportion of it happens. All right. Cheat and turn to page 6 of your guided notes. You're not finding it in your lab worksheet. You're trying to identify the difference in the bicarbonate ion formation between the plasma and a red blood cell. In the plasma, there is no enzyme used, so we said it just happens how? Slowly. What'd you say? Spontaneously, right? In the plasma, this reaction can occur spontaneously, and it is more slow than the other option. Conversely, bicarbonate ion may be formed in our red blood cells. What was different? Something called carbonic anhydrase, which is a what? An enzyme located specifically in our red blood cells. So bicarbonate ion can be produced, and what we're adding is either in the plasma spontaneously or B in the red blood cell under the use of carbonic anhydrase. That enzymatic catalyzed reaction is summarized on page 5 of your guided notes. What happens is the bulk of your carbon dioxide will actually diffuse into the red blood cells because they're so numerous in our circulatory system. And then once inside of the red blood cells, clearly some could bind to hemoglobin, but the bulk will interact with carbonic anhydrase to form something called carbonic acid, and this H2CO3. So obviously, what else do we need besides carbon dioxide? And carbon dioxide and water, yes, energy is in there as well. The reactant, though, would be carbon dioxide and water to form carbonic acid. And carbonic acid is described as being fairly unstable. And so once it's formed, depending on the direction which the reaction is going, we're studying it this way, it can catabolize into bicarbonate ion and hydrogen ion, which we learned bicarbonate ion is much more wet in our plasma. It's more soluble than just molecular carbon dioxide. Okay? You may notice as well the reactions are what? What do these arrows mean? They're reversible. Okay? And so I could just as well take bicarbonate ion and hydrogen ion and form carbonic acid, which we dissociate into water and carbon dioxide. And that's exactly what happens near the pulmonary capillaries. It's very much reversible. All right, so once bicarbonate ion is produced, if it's made in the red blood cells, it actually doesn't stay in the red blood cells. It exits or diffuses out of the red blood cell. It's transported, probably a better way to summarize that. Change the word diffused to transported from the red blood cell. Because bicarbonate ion is charged and we're transporting it out of the red blood cell, and we really don't want to change the polarity of the cell, a negatively charged bicarbonate ion is transported out, and then to make up for the balance, a negatively charged chloride ion is transported in. Okay? And as noted here, it's called the chloride shift. So send out negatively charged bicarbonate ion and then transport in, send out bicarbonate and transport in chloride ion. And we should maintain neutrality of the red blood cell if it's happening at the right rate. Does that make some sense? So our bicarbonate ion, no matter where it's made, is going to end up where? Where is it? In the plasma. Okay. Whether it's made in the plasma or made in the red blood cell, it's going to end up in the plasma, where we said it's much more water soluble. Okay. These forms of transport of oxygen and carbon dioxide between the pulmonary capillaries and the systemic tissues is summarized in those two images on page 6 of your guided notes, as well as basically what you did for handout 6, okay, where we looked at the pressure gradients. And so you can add a lot of detail in here. You can add the pressure gradients. You can add the forms of transport. Okay, there's a lot of detail that can be taken from this particular image. So first thing is, let's locate where we're at. We're at a particular tissue, okay, brain, skeletal muscle, stomach, whatever. And then, so we can see the tissues on the far left. The blue is the interstitial fluid, and the right is our blood plasma. So this image kind of plays out on you a little bit. So what I'd like for you to do is where you're separating the plasma and the interstitial fluid, just draw some capillary cells. And you can make them porous like we did in chapter 14. So just draw some capillary cells to separate the plasma from the interstitial fluid. You can see the directionality of the gases. At our tissues, carbon dioxide is being what? All right, what I want you to do over the evening, I want you to add the pressure gradient for each gas, for oxygen and carbon dioxide, and then identify the modes of transport for each carbon dioxide and oxygen. On this image, which is at your systemic circuit, and then the next image, which is at the pulmonary circuit. Okay. So you're adding, I'll come back to that text in a minute, but between these two images, the second image is an alveolus and a pulmonary capillary. I want you to as well reiterate those pressure gradients and summarize the modes of transport for each gas at the tissues. And you can use the text, what we've been talking about the last 30 minutes, and obviously handout number 6. If we're to back up and fill the text in the middle, we already are familiar then. The gradient is reversed. Any bicarbonate ion, we don't exhale bicarbonate ions, so it needs to get converted back to what? What do we need to convert it back to? Carbon dioxide. Okay. And so it can happen in one of two locations in the plasma or our red blood cells. If it goes in the red blood cells, we're using an enzyme, and it's the same enzyme, carbonic anhydrase, can take bicarbonate ion and hydrogen ion, form carbonic acid, which then dissociates into hydrogen ion, or excuse me, water and carbon dioxide. If I'm adding bicarbonate ion into the red blood cell, what should I take out? I'm at the pulmonary capillaries, and bicarbonate ion is transported into the red blood cell. What should I take out of the red blood cell? Oxygen at this point would actually be moving into this cell. Bicarbonate, if I'm putting bicarbonate ion into the red blood cell, chloride ion. Okay, remember? If I took bicarbonate ion out, I put chloride in, and then reverse it. If I put bicarbonate ion in, I should send out chloride ion. That is called, move back. Where I have it? Well, do I have it on here? On your image, I want you to circle this. It's called the reverse chloride shift, where if bicarbonate ion diffuses or is transported into the red blood cell, then chloride ion is actually transported out to reverse the process, to release carbon dioxide. We're to fill in our text. We're just reiterating, we said the process is reversed. Bicarbonate ion moves in, reverses back to release carbon dioxide and water. And then it's the carbon dioxide that we actually exhale from our lungs, not the bicarbonate ions. Can you see that reverse gradient? How carbon dioxide moves from the pulmonary capillaries into the alveoli. And that is carbon dioxide that we exhale, not bicarbonate ion that we exhale. And as well, the gradient is reversed for oxygen, load oxygen. the rest of chapter 17 talks about maintaining our blood pH, which we kind of dabbled in a little bit. Clearly, if our blood becomes too acidic or basic, we can bind or unbind particular particles to make sure our blood pH is at set point range. And it's just a balance between carbonic acid, bicarbonate ion, hydrogen ion to maintain our blood pH. If our hydrogen ion content becomes too high or our blood becomes a little what? Acidic. All I need to do is bind those hydrogen ions. Bind them with what? Bicarbonate ion and form which intermediate? Carbonic acid. And then conversely, if it switches the other way, if my blood pH becomes a little bit alkaline, so if my free hydrogen ions aren't readily available, carbonic acid can dissociate into hydrogen ion and bicarbonate ion. Okay, so it's just a nice, quick buffer system to make sure our blood is in set point range. Does anybody remember that set point range of our blood pH? Very good. 7.35 to 7.45 is the optimal range, right, for our body. And then if we fall below 7.35, we can bind hydrogen ions and bicarbonate ions. And then conversely, if we exceed that 7.45, we'll begin to break down the carbonic acid into hydrogen ions. What questions do you have over the respiratory system or the transport of our gases or their role in blood pH maintenance before we move to the renal system? All right, I'll conclude our reading here, our recording here, and then we'll go ahead and transition to